So let's get started looking at how the gonad develops. Now on this slide, we're looking at an embryo that we can say is about 5 weeks of development. And what we see on the back wall, the posterior wall of the embryo's abdomen, we see this pair of long mesodermal ridges that are called the urogenital ridges. And as that name implies, the mesoderm of that ridge will contribute to both urinary structures, specifically kidney-related structures, as well as gonadal structures. Now, the larger part of this urogenital ridge, the part that's shown in the lighter shading, is what's going to develop into kidney-related structures. So we're not going to talk about that right now. We'll come back and talk about that when we discuss the kidney. The smaller portion, indicated in the darker shading, is the part that will develop into a gonad. Now, even though it's this mesoderm of the gonadal portion of the urogenital ridge that will develop into a gonad, that's not where the cells that will actually become the gametes come from. Where those cells come from is actually out here from the wall of the yolk sac, outside of the embryo. Now, we'll discuss later on that the yolk sac is actually lined by endoderm, and it's cells from this endodermal lining of the yolk sac that will become the gametes. And what they do is they migrate from the wall of the yolk sac and to get into the embryo, traverse the umbilical cord, come into the embryo, migrate over to the urogenital ridge, and then invade or populate the gonadal portion of the urogenital ridge. Now, not only do those cells actually become the gametes, but they have another important function, and that is they induce this mesoderm to become a gonad. In other words, although this mesoderm, the gonadal portion of the urogenital ridge, is destined to become a gonad, it will not become a gonad. It will not differentiate into a gonad unless it is induced to do so by the migrating presumptive gametes. So we can say that when an individual has an absent gonad, the likely cause of the absence of the gonad was a failure of these cells to migrate into and induce the mesoderm to differentiate into a gonad. Okay, so now we know how we start to develop a gonad. The next question is, which kind of gonad will it be? Is this going to be a testis or an ovary? Or to broaden the question, will this embryo have a male or a female reproductive system? Now, the determination of whether the embryo will be male or female, of course, occurs at the moment of fertilization based upon what kind of sperm has fertilized the oocyte. And if it's a sperm with a Y chromosome, this will become male, and if it's a sperm with an X chromosome, this will be female. So we know that XX embryos are female, XY embryos are male. The important point being that what makes a male is the presence of that Y chromosome, and what makes a female is the absence of the Y chromosome. In other words, a female is not female because she has two X's. She's female because she does not have a Y. So that leads us to the question of what is it about the Y chromosome that causes maleness? So on the Y chromosome, there's a gene known as the SRY gene, and the SRY gene codes for a substance known as testis determining factor, which on this slide is illustrated as TDF. And in the presence of testis determining factor, that gonad will become a testis. In the absence of testis determining factor, it will become an ovary. In other words, the embryo is fundamentally pre-programmed to be female, and therefore that gonad is pre-programmed to become an ovary. And if you do nothing, it will become an ovary. But in the presence of testis determining factor, it will force it to become a testis. So that gives us the gonad, but of course there's more to having a normal reproductive system as a male than just having a testis. We need the other components of the reproductive system, the ductus deferens, prostate, seminal vesicles, and so forth. So where do they come from? Well, now that we have a testis, one of the cell types in the testis is the Leydig cell, and the Leydig cell produces testosterone and a derivative of testosterone, dihydrotestosterone. And in the presence of testosterone and dihydrotestosterone, they will differentiate all of the other elements of a male reproductive system. In other words, the ductus deferens, prostate, seminal vesicles, penis, scrotum, and so forth, as well as other secondary sexual characteristics such as body hair distribution, fat, muscle, and so forth. So because we have a testis, we have Leydig cells. Because we have Leydig cells, we have testosterone and dihydrotestosterone, and that gives us the rest of the male reproductive system. But because we said that this embryo is pre-programmed to be female, that means that it's pre-programmed to develop a female genital tract. In other words, uterus and uterine tubes and so forth. So if we want it to be a normal male, not only do we have to develop a male reproductive system, but we need to prevent the development of a female reproductive system. So that brings us to our next factor, which is listed here as MIF, which stands for Muellerian Inhibiting Factor, referring to the Muellerian duct system, which is where the female genital tract comes from. Muellerian inhibiting factor is produced by the Sertoli cell, which is another cell type in the testis, and Muellerian inhibiting factor inhibits the production of the female genital system. So now with all of those factors in play, testis determining factor giving us the testis, the testosterone and dihydrotestosterone giving us the remainder of the male genital system, and the Muellerian inhibiting factor preventing the formation of the female genital system, we now have a normal male with a normal testis and genital tract. In the absence of the SY gene, therefore we won't have TDF, therefore we won't have a testis, so we won't have those same levels of testosterone, we won't have Muellerian inhibiting factor, and so with the absence of those factors, we will have the pre-programmed ovary and female genital system. So because we see that this is a multifactorial process with several different elements of that process contributing to the normal development of the genital system, we see that we have the opportunity for errors in development that would lead to mixed gender abnormalities. So for example, if we had TDF and we had testosterone and dihydrotestosterone, but for some reason, let us say, did not have Muellerian inhibiting factor being produced, or we did not have the receptors for Muellerian inhibiting factor, then we would develop an embryo that would have a testis and a male genital system, but would also have a female genital system. And we can imagine lots of other combinations of errors that would lead to other kinds of mixed gender abnormalities. All right, so now we have the appropriate gonad, and now we have to see how those gonads will develop their gametes.